we start? Or?
I would go ahead and get started.
Okay.
People aren't going to stop until you start.
Okay. Can everyone hear me? Okay, so welcome back. We will be continuing this morning with the brain-body theme, and the first talk now is from Misha Ahrens from Janelia.
Hi, everyone. Thanks, Jennifer and Ron, for a great symposium, and thank you all for coming. I want to talk about brain-wide computation for flexible behavior and interoception. So the brain does many things, and it does them in parallel or in rapid switching, including learning about an animal's environment, learning about the physics of the environment and the physics of the motor system, forming memories and navigating, all the while controlling and sensing the organs that are inside the body. And these processes occur in parallel, and they often influence one another. So I want to present work by the people you see here, and I will also mention them as I go along. My lab works with larval zebrafish at about 1 week of age. And the reason we do that is because they are very small vertebrates that already display a variety of interesting behaviors at this young age. And there's a mutant that is completely transparent. We can express genetically encoded calcium sensors in the brain and other parts of the body, other types of fluorophores in other parts of the body to perform large-scale imaging. And shortly after I moved to Janelia, we worked with Philip Keller's lab to extend light sheet microscopy to be able to record from the entire brain at single cell resolution. So here you can see whole brain level, single cell level. We get more than 100,000 neurons like this. And we can do this during behavior by combining it with a virtual reality system that we developed earlier on, where there's a fish swimming through two-dimensional environments. These are visual environments. In this case, we're simulating water flow by moving the environment beneath the fish when it's not swimming, and it's stabilizing this visual stimulus by swimming against the current. The fish is actually paralyzed, and it's using its brain activity to control the visual stimulus and we're recording this from the motor nerves in the tail. So a fish exists in a sensory environment, as you just saw, that we simulated, and it interacts with the environment, with its brain. But the brain also interacts with a lot of other parts of the body, including the gut, and it's this recurrent interaction between the brain, the environment, and the insides of the animal that we're very interested in. So I'm going to tell you about 3 projects: navigation, and navigation with energy preservation, and interoception. So in a recent study on navigation, we often frame our biological questions in terms of the goal of the system, the algorithm the system uses, and the biological implementation of this algorithm. In this case, the goal of the animal that I'm going to show you is we call it positional homeostasis. This is work by N. Young, a very talented postdoc in the lab, and collaborators. So here you see a fish doing a behavior that is quite well known. It swims against the current. One interpretation of this behavior is that the fish senses the direction of flow, swims against it in a very feed-forward way. So it simply responds to an immediate stimulus, such as the visual flow of the world. Another interpretation or hypothesis of what the fish might actually be doing is that it measures the displacement here by integrating the velocity signals over time and correcting for displacements that may take much longer than an instant. And we found that this is actually the algorithm that most closely describes what the fish does. And we were very interested in this displacement term because one of our interests is in memory. So here you see the behavior of the animal in virtual reality. So the animal starts in an initial location of its own choice. Then it gets displaced by a virtual flowing current. to a new location where it gets clamped for a while, and then we give it a chance to correct for this during a swim period. And you can see that this animal swims back towards its initial location, and it's quite accurate at this. We wanted to know where this location signal is represented in the brain, so we performed whole brain imaging during behavior to get to cross scales, as is one of the themes of 4DCP, in search of this displacement signal. And we found it by simply correlating the location of the fish and neuronal activity throughout the brain. So this was an unbiased search across the entire brain, which yielded a collection of brain areas that contained positional information. And this, here you can see these brain areas. We had to invent a new name, slow-mo, for one of them, because it didn't have a name yet. We have yet to find out its homologous brain area in mammals. And from this, we can start building a hypothesized circuit diagram where we can look at activity in each of these areas, because this is all based on whole brain imaging. For example, in area slow-mo, we can see a representation of the location of the fish represented in different cells that perform positional integration. And looking at the population, we can now embed this in a low-dimensional space to track the location of the animal using network activity. If you ablate this area, then animals cannot stabilize their location anymore, so they lose the ability to form positional memories. So here we connected the goal, the algorithm, and the neural implementation, and now we could ask, how does this work when the animal also takes into account what's happening in its interior? And so what happens if we expose the animal to the same kind of challenge, but we also ask, how does it deal with energy preservation? And this is still all visually driven, but we asked, what happens if the animal minimizes displacement while also trying to minimize energy expenditure? So this is reminiscent of what the squirrel is doing, is trying to get to food, but the pole is lined with soap, so it quickly gives up. And the reason, most likely, that this behavior has evolved is because if it wouldn't do it, it would run out of energy and maybe hurt itself. And we implemented a very similar behavior for larval zebrafish, where they perform positional homeostasis that you just saw. They swim against a current, but now, because this is all in virtual reality, we can manipulate the transformation from motor output to visual feedback and make it impossible. So this is now impossible positional homeostasis, where the animal tries but doesn't manage. And what it does is what we would also do, it gives up, stops trying. Half a minute later, it will start trying again, and it will cycle between active and passive states. Now, we searched the brain for neurons that drove this passive behavioral state, and we failed for almost a year. Then we realized, also in the spirit of 4DCP, actually there are multicellular interactions in the brain that involve other cell types, not just neurons. In this case, we included radial astrocytes. So now you see a two-color recording with neurons on top, radial astrocytes at the bottom, and behavior at the very bottom. And we found that radial astrocytes are actually the cells that integrate the futility signal and then modulate neural networks to then bring the animal into a very different behavioral state. So here is a summary of the network. This is all in the brainstem, where there's a transformation, a handover of information that starts with neurodegenerative neurons, gets handed over and integrated over time by radial astrocytes. And the radial astrocytes, a glial cell type, then communicates back to neurons, in this case GABAergic neurons that inhibit future swimming. So here now, or two behaviors mediated by two different, very different classes of cells. And we could then finally transition to asking, how does this now interface with the control and sensing of organ systems? How does this all unfold together? So this is the final part of the talk, where I'd like to talk about interoception. This is a new research direction that we started fairly recently. This is work. The part of this work I'm going to show you is by Wei-Yu Chen, Benjamin James, Virginia Ruth, and James Fitzgerald, and also collaborators Manish Sahani, Florian Engert, Mark Fishman, Samba Shiva Banana, and Luke Davis. This is all preliminary and unpublished. So this is the the vagal sensory system in larval zebrafish that you heard about in previous talk in mammals. Zebrafish also have this system here. You see the vagus nerve, the nodus ganglion, that connects various organs, such as the gill, heart, liver, pancreas, gut, kidney, to the brainstem. This is another view of the sensory vagus. This is the motor vagus that is able to influence the dynamics and physiology of organs being controlled by the brain. We were interested in how the brain senses the interior of the body, and there are tools for this, but not quite as many as one would like. So it's useful to develop some more. Here we used light-activated approaches to release compounds in the gut that are photocaged. So the idea is that there's a photocaged compound. We uncage that with UV light shown onto the gut with a laser. And so this now we can release acid or glutamate or glucose in the gut while imaging from the brain using our light sheet microscopy techniques. And so this is building on work by many people, including some which are here. And we first asked, how does the nodus ganglion respond, just to make sure this system works. And we found single trial responses that were robust in specific neurons. Then we could transition to asking, how does the entire brain respond? So here you can see an image of a movie of the entire brain with certain areas highlighted, like the area postrema, the parabrachial nucleus, and the nodus ganglion. There are relatively robust responses in multiple brain regions. There's always an analysis challenge because there are so many neurons in the brain. We have to make sense of this data and find low-dimensional structure and assign responses to specific regions. Ben James and Wei-Yu Chen worked together to do this. They developed Bayesian analysis methods where they combined regression with mixture models to map response types to specific brain regions. And this then generates functional brain maps where individual regions are highlighted here. These are just a couple of them. We're still figuring out what these brain regions are. Neuroanatomy in larval zebrafish is still under construction, or our understanding is still under construction, but putatively we can use tools like ZBrain from the Engert lab to map these brain regions. As you can see here, a summary of this approximate mapping. Now, one of our questions includes, how does this information gets transferred from the gut to the nodus to the hindbrain from there across the brain? So one hope is that we'll be able to use timescales of responses to try and make predictions about how this behavior is handed from one area to the next. And so we can map the time scale across brain regions. Here's a color map of different time scales across the brain. This is now a very crude mapping projection along the dimensions, except for the longitudinal dimension. You can see there are specific regions where there's a more sustained response and others where there's a more transient response. Here are averages and individual neurons showing these different time scales. And this is one question we're asking: how does timescale respond to function, and what can timescale tell us about inter-area communication? Another one is how do these different compounds get coded in individual cells in different brain areas? And we, as an example, we took glutamate acid and asked, are there neurons that respond to one or the other or both? And this is inspired by work from the Liebers lab. Here, we asked this question in the nodus ganglion, where you can see that there are some cells that code for one or the other, and some cells that respond to both. We could do this now across all the other brain areas where we see similar patterns, but in some areas there's a bias towards one type of representation versus another. So where we're headed is to try and integrate these different phenomena, interactions between the brain and body physiology and behavior. And to close, I would like to thank the people who did this work, especially in Young, Martin Svart, and Zicheng Wei for positional homeostasis, Yumu, Davis Bennett, Mikko Robinov, and Sujata Narayan for the work on giving up, Virginia Ritz and Wei Yu Chen and Ben James for the work on gut interoception, and James, Manish Florian, and Mark Fishman, collaborators and co-advisors. And also thanks to everyone else at Janelia who's worked with us. I have 20 more seconds, but I'm going to say thank you very much for listening to me.
All right. Thank you, Misha. That was incredible. 3 stories in under 15 minutes. So here, I'm just going to toss the catch box.
That was a beautiful talk. Question related to that first vignette. Are you trying to figure out how fish compute their depth by chance? You could imagine using your system and changing the hydrostatic pressure and seeing what circuits are correlating.
Yeah, that's a really great question. We thought of looking at more dimensions. So there's also the lateral dimension, the rotational movement. Depth would be really cool to look at, and I think looking at pressure would be a great way. That's I'm guessing the primary cue. I guess one could also look at vertical, just simulating displacements in the vertical dimension using a visual stimulus that goes up and down as a first pass.
At this stage in development, do they have a developed swim bladder by chance?
Yeah, they have.
Yeah, so it may be the case that is ultimately what's going to provide the signals for computations.
The pressure sensed by the swim bladder would be a good cue. Yeah, thanks.
Go ahead.
So, really great talk. So my question is relating first and last part of when fish has this passive futility or futile passivity, I forgot which way, when they give up. If they have to swim towards food source and unable to move, and then they give up. And then if you modify their energy budget status, like make them hungry, would they go longer? Like that squirrel, would it continue going if it was really hungry?
Yeah, that's exactly the kind of question we're starting to work on now. So it's an excellent suggestion.
Question from Veena Prahlad. Do brain areas like slow-mo change if the fish lacked a fin, i.e. its definition of its own edges changed?
If the fish lacked a fin?
Yeah, so if the definition of its own edges changed, do brain areas like slow-mo change?
We don't know yet. We can manipulate the... I don't know how animals represent their own shape. But one way in which they can sense aspects of their body is through motosensory gain. So how much do they move when they swim? And we can manipulate that. And there is a dependence of slow-mo cells on that. So for example, if you go to the zero gain scenario, then slow-mo cells get down-regulated. So there is an interaction between body physics and slow-mo.
Misha, this is really cool. I have a technical question for the UV-induced cage system. It's really cool, I think, that method. So what can be caged? What stimuli can be caged in that system?
Yeah, that's the only, we're aware of only a handful. So we've done protons, glutamate and glucose. There are, I think, also caged other compounds of interest. But not everything. So we're still our goal is to expand it to several more interesting ones to try and tile at least a bit of the chemical space.
Go ahead.
Beautiful system. I had a question about the whole brain imaging upon gut stimulation. So gut hormones has been shown to be extremely unstable in vivo, and it's been really hard to track which neurons in circumventricular organs could really respond. So do you have a sense of all the circumventricular organs that you could image, which one of them could respond to the stimuli? You talked about erythropoiesis, but...
Yeah, so organs, we're currently focused on brain responses. You're talking about whole body responses? Or?
No, okay. So I don't know zebrafish anatomy well, but in mouse brain, there are circumventricular organs that lock the blood-brain barrier where they could sample gut hormones in the blood circulation. And I was curious if there's an analogy in your system, and if so, how much of these neurons, which brain regions could respond directly to humoral pathway, I guess.
Yeah, so I think there's a lot of there is a lot of homology. Definitely the area postrema can sense the blood, contents of the blood. There are some other areas in the zebrafish brain that can also do it. I don't know if they exactly map onto the mouse areas, but many of the structures, hypothalamus and everything, is conserved. So I would imagine that's also conserved. But I don't know how much is known in zebrafish, but we don't know at the moment in the lab. I think it's very worth looking into. The time scales we're focusing on are relatively short, so this is one reason that we think looking at hormones would require kind of looking on longer time scales as well.
Okay,
it would definitely be interesting to do.
Thank you.
So Beth, since you're standing, let's ask a question, and then Anoj, you want to set up?
Yeah, I'll ask. My question is more of a next step question. It ties together the last part of your talk, which was extremely exciting, with the imaging of the gut brain, with the cells that are responding in the brain. And so I wondered, given your previous work and the tools that you and others are developing for sensors, what about the glia in the brain? Could they be interconnecting the response of those different regions of the brain, different circuits? And are you planning on looking at that?
Yeah, I very much so. So I've been very much asking Wei to do these experiments, and I believe she has done them. But we have yet to analyze that data. But that's definitely of huge interest. Thank you.
Excellent. All right. Thank you, Misha. Okay, so our next speaker and last speaker of this shorter session is Anoj Ilanges from Janelia.
Okay. Is the mic? Okay. Okay, thank you to the organizers, Ron, Jennifer, Janine. It's great to be able to give a talk in such a sort of diverse group, looking at so many different things. I've only been at Janelia for about a month as well, so this talk will really focus on my work that I did when I was a graduate student at Rockefeller in Jeff Friedman's lab. And there, what we were sort of interested in is getting a better understanding of how the brain controls some of the behavioral responses that happen during infections. So, what does that mean exactly? So, usual day for us is we're, really excited to get to lab, hang out with our, lab mates, do a couple of experiments, grab maybe lunch together. And then, this is something that we're sort of hyper aware of now. Sometimes we feel sick, and, what does that mean? When we feel sick, we're, not so interested in hanging out with our friends anymore. We're not hungry. We might have a fever, and all of a sudden, our bed seems like, the most exciting place to be. And what's really interesting is that this is a really sort of stereotyped set of changes that happens amongst different people during infections, and we're really good at actually recognizing this. And, a work by a veterinarian in 1988 recognized that this is actually something that's fairly conserved amongst almost all animals. And he put forth this idea that actually this is not a result of sort of damage caused by whatever infection we have, but is actually a response induced by the host itself to better survive infection. And a large degree of work even before that, and as well as more recent work even by Ruslan's lab, have shown that some of these responses, such as the anorexia, is actually really important for helping animals to survive during infection. So if you force feed animals that have a bacterial infection, so basically sort of abating some of the anorexia that's there, animals will have an increase in mortality, really indicating that some of these responses, again, are real adaptive changes. So since then, there's been a lot of work sort of looking at how this state is induced, and really beautiful work has shown that it's cues produced by the immune system that interface with the brain, either through circulation or through the peripheral nervous system to enact this state. But what was less so understood is sort of what populations in the brain are actually mediating these really stark changes in behavior that seem to happen. And that was sort of our goal at the onset of this project. And for this work, I received help from a really talented graduate student that I was training at the time, Rainey, as well as a research assistant, Jordan. So to start looking at sickness behaviors, we wanted a really robust and consistent model. What we decided to use was lipopolysaccharide, or LPS, a component of gram-negative bacteria that is really well sensed by our innate immune system, and allows us to sort of induce a bacterial-induced infection state without a live infection causing damage to the body. So what we wanted to look at are some of these responses, and we used a home cage system that we developed that allows for really high resolution measurements of food intake, water intake, and movements, the behavioral parameters that we really cared about. And then we started to ask the question, what do different doses of LPS induce in terms of response? And what we found is that there's really potent responses to different doses of LPS, with reductions in food intake, water intake, and movement seen amongst them all. But we could see that, as with other sensory systems, there is a scaling of the response due to sort of the dose of this inflammatory stimulus, as well as there being coordination in sort of the induction and duration of these responses, indicating to us that this was sort of a really bona fide program as opposed to individual responses. And we looked at some autonomic changes, such as body temperature, and what we found is that there are really strong responses in that, but the time scale of that doesn't seem to be coordinated with the behavioral changes, indicating most likely differential mechanisms of control. Now, with this system in hand, we could start to ask what's going on in the brain of these animals to induce such a response. And to do this, we used a sort of proxy for neuronal activity called FOS. And we could stain for this sort of brain-wide in animals using tissue clearing and mapping to ask sort of holistically throughout the brain what's going on. And here is a video of a mouse that was injected with LPS 3 hours before. And what you could see is that there are really tight nuclei of activity throughout the entire brain, indicating to us that there's quite a lot going on in these sick animals that actually don't do much when they're sick. And we could really quantify this across different times to start to look at sort of the global response. And what we could see is that there's sort of a peak in the response at 3 to 6 hours that abates by a day, fairly well correlated with behavior. And what we were really excited by was seeing that 2 regions in sort of in the brainstem, the nucleus tractus solitari that we've already heard about, the NTS, and area postrema, were activated early and had really potent sustained activation throughout this response. And the reason why we were activated by this is that a rich set of literature has shown that this area can interface with circulating factors in the blood due to a permeable blood-brain barrier, and that it's the entry point for the afferent vagus into the brain. So now with this information in hand, we did do some functional tests to show that this region can most likely be involved. But I'm going to move ahead to talking about sort of the molecular characterization that we did to sort of identify which cell types in this region might be mediating this sickness response. So to do this, we use single nuclei RNA sequencing. And we first did sort of a baseline sequencing of this region to get an atlas of the general types. And then we used an activity-based sequencing with the TRAP2 mouse line, which allows for CFOS-based tagging of cells so that we can express a nuclear GFP marker in LPS-activated neurons, then go cut out the region, sort out these cells, and sequence those cells specifically to ask which cells are specifically enriched as LPS activated. And this is just the baseline sequencing, and as has been reported by many others, there's a rich repertoire of different cell types in this region. And now, when we map the LPS activated cells onto this sort of baseline set, what we could see is that the sickness response induced by LPS activates sort of a range of different cell types. It's not a singular type in this region, and that there's excitatory and inhibitory cells activated. And we did subsequent experiments to show that they were the excitatory cells seem to have the behavioral phenotypes we cared about. So I'm going to go and show some of the characterization we did for enriched excitatory cell types from our data set. And what you could see is that there are different cell types enriched in this region during the LPS response, but the ADCYAP1 neurons were the most enriched cell type across the NTS and AP collectively. So now that we had a cell type in hand, we could start to ask how do different activated cell types contribute to the overall sickness response. And to do this, we can use Cree lines that target these cell types and introduce an AAV virus specifically into the NTS AP region to deliver Cree-dependent activating dreads, or GQ dreads. And so we can ask what happens when the animals aren't sick, there's no inflammatory response going on, but if we activate these neurons, do they act sick? And when we did that, what we found is that remarkably, just activating this one population of neurons in the brainstem is able to induce really potent changes in food intake, water intake, movement, as well as thermic changes and body weight changes that have the same sort of features that we see from LPS, which is a really a strong magnitude of response, as well as a long duration of the response itself. So while we did this for this one type, we did notice again that in our sequencing, it wasn't just a singular type of neurons that were activated. So we decided to ask, what are the other neuronal types in this region, which are somewhat overlapping also with these ADCYAP1 neurons contributing to the response? So we activated each of these different types to see if we could see segregation of behaviors or specialization in any way. And what we found generally is that most of the neurons in this region induce sort of coordinated changes in these different parameters, and that the main difference between these different types is the magnitude of the change induced. And this indicated that at least at this level and at this region, there isn't segregation of the food intake, water intake, and movement at the cell types being activated by LPS. So while we were able to show that these neurons are sufficient to drive the response, it doesn't tell us necessarily that these are the ones physiologically mediating the response. So then now we can ask the question of, using the same sort of system, can we make animals sick with LPS and activate these neurons and ask what happens to their behavioral response? When we did that, this is a little busy, so I'll take you through it. When we have animals that have nothing inactivated but are given LPS, we see a really strong sickness response. And in this yellow line, you could see animals that have been just given, saline, nothing's wrong with them. And this is sort of the baseline food intake in this case. Now, if we inactivate these neurons and make these animals sick, we could see that we can rescue about half of the deficit in this case in the anorexia, indicating to us that, and this held true for the water intake and movement, we did not see any changes in thermic control and with body weight as well, indicating to us that these neurons are indeed important for mediating the full magnitude of the sickness behavior response, and that there's most likely other regions throughout the brain contributing for sort of the full magnitude, which makes sense from the different populations we saw active. Now, if we test other lines that we saw, we're able to induce some level of this sort of sickness response. We see that they are unable to block the actual induction of this response, indicating that the ADCYAP1 neurons are most likely the main player in this response for this region. Now, the goal at the onset of this project was to really identify a population for sickness behavior control. This is really only the first step in trying to understand what's going on in the brain during these really complex infection states that require a lot of interaction between the body and the brain. And sort of there's some proximal questions related to at least how this is happening at sort of a more behavioral level. And some of these are, how are these neurons being activated? Are these peripheral cues from the blood? Or is this brought in by the afferent vagus system or the spinal system? What are the other activated populations contributing? We see quite a lot of activity, but this is just one region in this whole network of activity, and work from other labs is also starting to really look at this question. And then finally, can we sort of identify an effector circuit responsible for the output of this? Can we see that if we go downstream of these neurons, if we could see segregation and specific control of the specific behavioral changes that we see, giving us a better idea of how to actually modulate the system? And while the focus of this project has really been on sort of this behavioral changes that happen as a result of infection and immune signaling, there's a lot more that goes on during infection. And there's really potent changes in autonomic function as well as systemic metabolism that happen during the state. And this is also derived predominantly through immune signaling, and work has shown that the brain mediates a large part in this process. So going forward, I think we really want to deconstruct this axis in terms of what signals from the immune system are causing what changes in the brain, what these changes in the brain actually mean, and why are they so potent. And then finally, how is the brain sort of controlling this top-down control of physiology during this state and coordinating amongst behavior, autonomic function, metabolism? So this is sort of the core direction for the future. And for this work, I'd like to thank Jeff, who's a phenomenal mentor during my graduate work, as well as Rainey and Jordan, who helped in these projects. I'm working with Misha on some of these questions in zebrafish as well now, and we're looking for additional people to come help out and work on some of these problems. So with that, I will take questions.
Do you want to use the microphone, or? Thank you.
Yeah, Jim Wells, that was a very nice talk. I wonder, in thinking about the way the immune system is speaking to the brain, cytokines are an obvious messenger that they use, and I wondered if you've looked for those receptors for specific ones, and maybe they go through glia to the neurons themselves.
Yeah, so I think this is a really I don't know if I have the data. So we do see the expression of some cytokine receptors in some of these neurons in this region. We do also see activation of glial cells during these sort of really potent sickness responses. So I would think that a lot of the sensing is actually probably happening directly through the glial cells in this region, and are being probably transmitted to the neurons to produce sort of long activation. And this is a question that we're starting to, I think about using zebrafish, because the imaging there really allows for sort of uncoupling the neuronal responses as well as glial responses brain-wide. So it's something we're thinking about. But yeah, neurons I think can respond, but I think glia are actually better set up probably to respond and really accentuate this.
That was a really cool talk. Can I ask 2 questions? First question is there a role for GDF15 in GFRAL in this response?
Yeah, so GDF15, it's been shown that it's not as required for sort of the behavioral responses, and even the anorexia, that's not sort of the potent driver of that. But really nice work, actually, from Ruslan shows that it's involved for a lot of the metabolic adaptation during some of these states in terms of providing different organs, different fuels and stuff as a sort of adaptive physiological.
So does GDF15 not go up following
So
LPS?
GDF15 does go up, but it doesn't seem to be controlling sort of the behavioral aspects as potently as it's actually controlling some of these really interesting metabolic switches. But yeah, GDF15 is involved, and GFRAL is only expressed in this one part of the brain area, postrema. But we don't see that that's sufficient to sort of, if you get rid of that axis, it doesn't change the behavioral response during infection.
And then the second question is, I don't know if it was your work or the classical work, but when you force feed the mice that don't want to eat, what's the cause of death?
Yeah, that's a really good question. Ruslan could probably answer that one better, because he worked on some of this for bacterial versus viral infection. And from what I remember, and sort of the most convincing of this was sort of, it leads to sort of imbalances in glucose being shunted to like the brain and leading to seizures and stuff in some of these cases, indicating that sort of the body isn't really set up to handle these sorts of influxes.
So it's not just a simple propagation of the infection, but it's actually something else.
Yeah, it seems to be more of it's not a resistance-related phenomenon, it seems, where in terms of like controlling the actual infection itself, it seems to be something about this sort of idea of tolerance and how our body actually is set up to sort of survive the damage caused by infections, and our immune response provide the necessary fuels to the right places and sort of clamp down certain
But the
settings.
ultimate cause of death is?
I think it was seizures and the glucose amount.
Neuronal damage.
Yeah.
In the presence of glucose and inflammatory signal, and it's rescued by.
Yeah.
So glucose kills because it's suppressed.
Okay. Last question. Go ahead.
Yes, some of these behaviors are reminiscent of a depressed person. How you feel that these a circuit that you're studying is related to that as well? In the case of the depressed person, everything comes from the top, the brain, and goes into the body. And in your case, it's more external component affecting the circuit.
Yeah, so I think some of these axes have been implicated in sort of depression, and there's even been really nice work showing that there are correlation of some cytokines in circulation and some of these really in major sort of depressive disorders. So I think there is this idea that some of these circuits play a role. I'm not sure if it's exactly this one or sort of a subset of this sort of global circuit that we're talking about. And I think that this is where it's probably really important if we break down some of these complex states into more cytokine-induced states, we might get a better idea of which ones might be mediating some of that.
Okay, I think, Janine, you have one question?
Yeah, just one last question that came in from Ulrike Heberlein. Are there candidate brain regions for subsets of sickness behavior?
Yes, so there's been some work from Catherine Dulac's lab, for example, that shows that maybe fever is controlled by this preoptic area that's really well known for sort of thermal control. I would imagine that if you look at some hypothalamic regions, you might potentially see that they might be more involved in feeding for example. But as of now, we don't have a super clear idea of sort of the segregation of roles in the circuit, and I think that's a really important question going forward.
Thank you.
So I think we should wrap up. I want to make sure everyone has time to eat lunch. So you have exactly 50 minutes. And so please be back here at 12:30. And thanks. Let's thank all the speakers.